Yes. Hello everyone, I am Harsiddhi, President of Tamil Nadu Node for the International Society of Computational Biology Student Council, Regional Student Group India. And today we are honored to host Dr. Anandhi Iyappan, Data and Metadata Standards Officer from EMBL Heidelberg for our third web lecture, Bridging Genomics and Openness, Data Governance on Lessons from GHG A, in collaboration with Sri Ramachandra Faculty of Engineering and Technology and the Department of Bioinformatics. Welcome everyone. Before we proceed to the event, I would like to give a brief introduction to ISCB-SC. ISCB-SC RSG India is your local window to global opportunities in computational biology. ISCB is a global organization based in the USA, and it drives computational biology forward through research training, outreach, and community building. The ISCB Student Council, started in 2006, nurtures the next generation of computational biologists with RSGs in over 26 countries. RSG India connects and promotes a strong nationwide network of computational biologists linking local talent to global opportunities. We proudly collaborate with premier institutions including Mahindra University, NIT Calicut, Manipal Academy of Higher Education, and our very own Sri Ramachandra Institute of Higher Education and Research in Chennai. Here is the RSG India Core Committee from 2025 to 2026, supported by faculty advisors and visionary leaders across India. RSG India has successfully organized impactful events in the past, fostering collaboration, learning, and innovation in computational biology. Here are the pictures from some of the events organized from 2023 to 2025. You can reach us through our website or connect with us on our social media platforms. Now, I would like to call on Abdul Aziz, Secretary of Tamil Nadu Node, to brief about the student initiatives at SRIHER.
Good evening, everyone. My name is Syed Abdul Aziz, and I'm the Secretary of the SRIHER node of ISCB RSG India. And I'd like to take a moment to introduce the SRIHER node of ISCB RSG India. We are a student-driven community with a vision of creating a vibrant platform for aspiring computational biologists and bioinformaticians. We were established this year in 2025. And our goal is to create a platform where bright minds can collaborate with researchers and professionals here in India as well as throughout the world. And our aim is to create workshops and events and webinars such as this one and on-campus events to encourage learning and collaboration throughout. Next, I'd like to introduce as the team behind this effort of creating these events. We are both from undergrad and postgrad level and across all of the years. So this is a picture from our very first introductory event held on campus here on Ramachandra. And a special mention to our advisory board consisting of our very own faculty from the Department of Bioinformatics, Dr. Dicky John Davis, Dr. Abhinand, and Dr. Arundhati. Without further ado, I'd like to pass it on to Dicky sir to introduce our guest speaker this evening. On to you, sir.
Yeah. Good afternoon, good evening, everyone. It is very nice to introduce our alumni, Dr. Anandhi Iyappan. She was the batch 2007 student, and she was a third batch student, and presently she is a data and metadata standard specialist with over a decade of experience in bioinformatics and semantic technologies. She works in EMBL, and she leads with the metadata and ontology integration of German Human Genome and Phenome Archive. she has a very open thing about the fair champion, and she advocates a fair and open science practice across Europe. And Anandhi holds a PhD in applied semantics. And when I looked into her PhD, her PhD was on conceptualization of computational modeling approach and interpretation of role of neuroimaging indices in pathomechanisms for clinical detection of Alzheimer's disease. She has published a lot of articles in the Journal of Alzheimer's, in Lancet, and many other journals, including the Oxford Bioinformatics Journal. Presently, the latest she's one of part of another repository called as Metalog, which is a repository for global metagenomic samples. Um, she has a lot of experience in data, metadata, and semantic works. I um, wholeheartedly welcome her for this one and to present her work. Welcome, Anandhi.
Thank you so much, sir, for having me here, and thank you for the organizers for giving me this platform. I'm sharing my screen, so just let me know.
Harsiddhi, close the screen.
Are you able to see my screen, sir?
No, not yet.
And now we can.
You
Okay.
can? Okay.
Yeah, now we can.
Okay. Sir, so anybody, if they have any questions in between, they can stop me. I'm not able to see anybody, so you just have to just unmute and ask questions. Yeah, and also you can take the time at the end and stop me anywhere in between as well. Yeah. So with this, I would like to give you a
Anandhi,
journey.
put it in slideshow, ma.
It is in slideshows, sir.
No, but we are seeing it.
Are you, because it's a full screen slideshow, that's what I've enabled.
No, we are seeing the other one.
Maybe you're using the dual screen one. I think. So
What is it?
you're using the
Okay.
dual screen, dual monitor.
Yeah, I'm just
You
give
have
me a
to
second.
share the
Okay.
second screen.
Yeah, I'll just. Okay. 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 Now?
Yes, ma.
Now
It's
okay. Okay.
perfect.
Perfect. Awesome. Thank you. So I just wanted to give a small overview from the time I've come to Germany, which was in 2011. I have really seen that the amount of work that came in terms of genomic projects were quite huge, ranging from the PQ or the 6G project in 2008, ICGC, to where we are now with the Human Cell Atlas project. And it was also thought very deeply that all of these projects were generating a lot of data sets for genome, human genome, or a non-human genome. But Germany basically lacked a strong infrastructure for archiving and sharing human genomics data. And that led to the vision of this project that we wanted to create a platform where people from the genomics community can share their data with, because we have a strong ethical legal framework put together, it makes it easier for them to share human genomic data, which seem to be very much distributed across other centers in Germany. So this bridge that you see here is what we want to be as a project, to be a bridge between the research institutions and in some way also with the healthcare, where we get the actual data from. And we want to be a bridge between these two communities, which enabling responsible data sharing and data management as well. So the idea of this project is to establish a nationally coordinated interdisciplinary infrastructure for integrating genomic research and healthcare. Also, the focus on this project is mostly on the secondary use of clinic omics data in research, which of course will lead to biological discovery and also to replicate findings that show validity as well. This project is funded by the NFDI, which is the National Research Data Infrastructure Initiative, which funds projects like this, especially on the FAIR aspect of research data. The project offers about 900 million euros, and it's distributed over 10 years. And it supports various projects under this wing of NFDI, such as NFDI for health, NFDI for microbiota, for bioimage, various research communities with similar intentions of wanting to do responsible data sharing. And we as a community, the GHGA, is strongly interlinked with all of these NFDI consortia as well. So the setup of this consortium is, as I said, it was funded in the year 2020, and we are as one of the first 5 NFDI consortia which were given funding for. This project is very huge, and therefore we have NETs, or the genomic research centers, all over Germany working in this particular project. We have about 7 data hubs distributed across Germany in cities like Tübingen, Kiel, Dresden, Heidelberg, Munich, and Berlin. All of these data hubs are also making use of a national cloud infrastructure, which is called the DENB cloud, to share or to store the data. And this service is actually available free for most of the German institutions to store data, and they write research funding projects to get access to this platform. Also, this project, about 15 million euros has been the funding over 5 years' time, and of course, it is also shared amongst the partner institutions. So I work with both the professors Oliver Kohlbacher and Jan Koppel, who are all based in the EMBL, and Oliver Stegle is also one of the board of directors, who's my direct supervisor in this project. So when we talk about human genomics data, what is really special about it, right? So why is it that we have to be so secure, and why do we have to think of various ways in which we think about responsible data sharing? Human genomics data is, first of all, multi-layered, because when you say omics data, omics consists of various layers such as genomes, the exomes, epigenomes, and transcriptomes. What differentiates genomic research and a portal that we are developing is the key connection, which is the clinical metadata. It's not just about sharing the research aspects of data, but also the clinical information pertaining to these research data, which makes it very challenging for a data service provider to think about data sharing. Of course, this gives a strong research opportunity for the community, but of course that needs to be balanced with strong controlled access measures that we need to take to ensure that the data is well protected. This project also deals strongly with contractual regulations, especially after the GDPR law came into place. So a lot of policies had to be put in place to ensure that we do responsible data sharing and de-identification of patients. And of course, it also has other organizational barriers when it comes to sharing data, especially from clinics of different regions. Of course, the research aspect also needs to be considered, because whatever we are doing is to foster the research community. So there are multiple stakeholders involved, making it more complex as a project. Currently in this project, we are focusing on rare diseases and cancer. And of course, in the future, we would like to expand to other disease modalities as well. So this GHGA project works as a federation concept. We call it as a federated concept mainly because it is also one of the first projects which is being designed on a national level, which is also being part of, and not just within Germany, but also within other European countries as well. So that makes it the first project to do so. How this project works is, as I mentioned to you in the beginning, we have about 7 data hubs, which are kind of connected to the genome centers who generate sequences, and they're analyzed in these data hubs. All these 7 data hubs are connected to the central data hub, which is the GHG A Central based in Heidelberg, which is also where EMBL is placed, where I am based. And this central EGA, central GHGA will be the source of contact to the external community, such as the European Genome Archive. And the European Genome Archive is also a federated node for other European countries, such as Sweden, Norway, Spain, Netherlands, to name a few. So it's quite complex, and we are also the national node for the European Genome Project as well. That's how we got into this consortium, and that's how we became part of this global, I wouldn't say global, but at least European network-wide community. So this project, as I said, it's very, very complex. So therefore, we have various work streams who are taking part. One is the architecture work stream, the other one, which is, of course, looking into the different services, infrastructure to provide to this data portal. The data hub or the stewardships who are looking into the contracts, the legal contracts, as well as the submission process of data that comes into GHGA. Metadata is where I am part of. I am the metadata coordinator of this project. My key role is to ensure that we design a data model pertaining to the community needs, and my role is basically to cater to all the needs and come up with a model which will support data sharing. We also have other work streams such as workflows, developing pipelines to generate, to reprocess the data that we get, LC community focusing on the ethical framework of the project, training community coming up with research training modules to reach to the research, to the wider research community, outreach to obviously promote what we do, and of course a strong project management team which looks into the overall aspects of the project. The project basically functions in 4 phases. We have already launched the first and the second phase, which is the metadata catalog phase and the archival phase. The first catalog phase was the first operational phase of the project, where we were basically trying to collect the existing resources that we had from the genomics community. We were just trying to set up a library which is compatible with the EGA, because we are the federated node of EGA, the project European Genome Archive, and therefore we had to be in line with their infrastructure. After that, we have recently launched the archival phase, where we are in a phase of opening our data model to the general public, where we will give the public to come, like just explore the data portal and request for data. Currently, we have just launched it a few months back. And the idea is we are able to deposit up to 500K genomes in the upcoming months, hoping that this will also make our project widely known to the bigger research community. In the second funding phase, which will start in next 3 months, where we are interested in also exploring the secure processing environment for the project. And also at some point, we want to come up with an atlas phase where we allow analysis to be done, visualization of the data, and also to perform other analytical aspects for research, which is something that we have planned for the second phase of the project, second funding phase of the project. So having given you the background of what this project is about, I will tell you what I'm currently doing in this particular project. I'm as I said, taking care of metadata. I'm responsible for designing the model for this project. So when we started, it was very important for us to not just think about the first phase or the second phase. We had to design the model thinking of, say, 20 years down the lane. If this project is still going to be alive, what are the measures that we need to take? So we had to do a lot of research on, because we didn't have a blueprint to come up with a design like this, so we had to really do a lot of groundwork. So the idea that we came up with was to have a structured approach in order to organize human omics data, complying to the FAIR principles. FAIR stands for findable, accessible, interoperable, and reusable. And based on these principles, we wanted to design this portal for data archiving and sharing. So the main components of this metadata model is obviously we're going to have a strong overview on what this model will capture. The second aspect is, of course, what kind of metadata we want to capture. That is also a big task because it is a very wide metadata is, first of all, extremely illustrious. So therefore, what we want to capture is something that was a big question that we had to address. Of course, we wanted to be a standardized platform, so we had to look into the existing concepts and standards related to designing this metadata model. And of course, we wanted to come up with a dictionary or a data thesauri to capture the different concepts, what they mean, the meaning, and how they are basically like a glossary for future purposes, and also to document the current resources that we have in the form of a data dictionary. I mentioned to you about the FAIR principles. The idea of FAIR principles is what the whole data management and data stewardship is encapsulated, basically. There's a very interesting article published in the year 2016 on what FAIR principles are and how they should be implemented. So those interested in this, I would recommend you to read that paper. So the idea behind FAIR principles is to ensure that whatever data is being designed and managed is, first of all, findable by other researchers. Second, accessible in order to make use of the data. Third, interoperable, which is if you have different platforms, how do you communicate between each other? So making it more interoperable. And finally, the most important one, reusable. So if the data is designed in such a way that it is strongly and well annotated, then it is going to be enabling easier reuse for diverse use cases as well. So the GHG A metadata model was built using a LinkML modeling framework. LinkML is a schematic modeling framework, which is highly rich with vocabularies and concepts. It is based on a YAML, yet another modeling language framework. And we wanted to have this model framework as a single source of truth to collect metadata. And we ensured that the schema that we designed should be self-describing, so no further interpretation is needed. It should be unambiguous, flexible, and expressive. The word flexible is very important because you will see in the upcoming slides why I'm emphasizing on a model being flexible. Of course, the schema should also be in a position to capture the different concepts pertaining to a certain modality. Say, for example, if it's cancer genomics, then what different modalities can be captured? The model should help us to capture those concepts. So we looked into various models, and LinkML seemed to fit the purpose for us. So it basically looks like this. So you can write it like a statement where you can capture as much information as you want into the model. And this is spread in such a way that you have a class concept where you can design experiments or samples, which are the bigger concepts pertaining to a certain experiment. And then you can add further attributes to the classes. Like if you have an experiment, you can have different features to it, like name of the experiment, type of the experiment, description of the experiment, and so on and so forth. And of course, you can also define the value, the data type, whether it should be an integer, or it should be a string, or it should be a float. That can also be designed, and it can be incorporated into this. So it helps us to think in terms of a biologist to design the model. So it made it simpler, and also for somebody who has no idea about data modeling, this framework is rather easy to adapt because it also prompts different values. So it's quite easy to use and build. So we went with this framework. So now I will show you the different phases of this particular model. The word flexible, you will see why, and why I emphasized it in the beginning. And also I wanted to mention that we could not design the model from scratch because, as I said, we are working as a node to the European Genome Archive project. So we had to stick to a similar structure that that project had. So we started from the schema that they put together on what concepts they had captured. So our core skeleton of the model design will look similar to what EGA had, but we will add more flavor to it. So if you see in the left-hand side is what you see from the EGA prototype, and what you see on the right-hand side is the prototype we had put together. So we got rid of a couple of classes like assay and analysis from EGA. Instead, we modeled them into a different concept called library, for reasons I can explain to you after the presentation, if time permits. And we also added more layers to it, like publication, capturing information about the member for provenance, in addition to the existing model from EGA. So the first model looked like this, which was quite, I would say, voluminous in terms of the different concepts we were trying to capture. So the model was designed again to not just think in terms of research data, but also in terms of the data governance on what kind of conditions in which the data was captured. So with this design, we started the first phase of the project, which was during the catalog phase. Soon we had to deal with a lot of trade-offs with the first design that we did, because the first question that we had to ask was, whatever design we are putting in place, can that actually balance between the data reuse and data protection? Was that model good enough? We had to challenge that a couple of times to see. And whatever information we are capturing, was it also being GDPR compliant? The second aspect, what made us rethink this design that we put together, was the granularity versus excess demand. As I said, metadata can be very voluminous in terms of the amount of data you can capture. So how much detail is too much detail, and how less detail, like how less information you capture is less. You know, less is more, or more is less, or more is more. It was very, very difficult, because thinking of a model design in terms of an ideal scenario, which means I would want everything to be captured. But when you think, what is the purpose for which this data model was designed, is to make the researchers to provide data to us in such a way that they can share it with others. So we don't want them to also be burdened by the amount of information we ask from them. So that was the trade-off we had to be dealing with. We were dealing with. And that led us to come up with a slightly different design of the model, which is called the modular version, which is different because we modularized the concepts into various, into various blocks. So it provides the user to pick and choose which module he wants to contribute to, and how much information he wants to provide. So this model was also well received, but it also came with certain limitation. The first limitation was that it was very much sequence experiment centric model. If you see in this model, you will not see experiment at all. You will see the module to be a sequencing module, because at that point of time, the amount of data sets that we received were all sequencing related. So we redesigned the model just to cater to the need for sequencing data sets alone. But later we realized that genomic experiments cannot just be sequence-centric. We should be thinking about other omics data types like proteomics or spatial omics. Then this model will definitely not be good enough to cater to those needs. And the next trade-off was, of course, as I said, the different communities means you will have different needs and different expectations. So when we challenged the model again, we realized that no, this is probably not the best way to go about. And currently, we are at the archival phase of the model, which looks like this, which is probably going to be the fixed version of the model going in the upcoming phases, mainly because we think we have come up with the most abstract version that we can. And it will suit to the needs of the communities who will be making use of this data portal. So what we did differently here was we tried to come up with a distinction of what we want to call as metadata itself, and how do we distinguish them, how should we distinguish them from what a researcher can provide and from what we can bundle and make a data set on our own in the data portal. So whatever the information we will be getting in terms of data from the data producer will be the research metadata, which will contain information pertaining to the individual, the sample-based information, experiment-based information, and analysis that they are performing. And the other aspect is the administrative aspects of things, which will be looking into the contracts, into the data access policies and the data access committee that should be responsible, including the person who's providing the data, what information they would like to give to the community. And all of those will be part of the administrative metadata. So this design basically was done, one, to simplify the submission process into our data portal. Second of all, helping the user to have a clearer way in which they can submit their data, which was also a complaint that we received from the previous users, that the model was a little bit difficult to follow because it did not have a clearer structure. So we came up with this design, and just to show you a little bit a bigger overview of what I mean by the archival version and the abstract version was, we tried to make this model be designed in such a way that it caters to different types of experiments, not just sequencing, but also to multi-omics, spatial, to name a few. So what we did was I've expanded the experiment aspect here alone to show you what we mean by this structure. So for us now, the experiment is an abstract class, which is, it will contain the minimal information you will need to know about the experiment. Like, for example, in an experiment, what are the most common things that you need to know, like title, description, type? That can be the basic information one can provide. Then we have a concept called experiment method, which is a subclass of experiment. And the experiment method here are the different types of experiment modalities, like omics modality, exome experiments, proteome-based experiments. And in each of these modality can be distinct, we can come up with distinct templates for each of these modality. Therefore, it saves the data submitter or the data provider in giving us with all the information that we ask them to provide. Instead, they can just say, look, I have a genome experiment, then I will only use the template pertaining to the genome experiment, and I will submit the data. So this gives the user the freedom, the degree of freedom to submit the data that they want to provide. And even from our side, it helps us to deal with use case-based submissions. And therefore, it was a win-win situation for us. And therefore, we thought this design would be the best design to go forward. So we also had to come up with clearer distinctions and definitions of what we mean by a GHG A metadata model. And when we say a research metadata, what do we mean by it? So a research metadata for us basically captures all kinds of metadata pertaining to an individual sample experiment. And it can also include information related to biospecimens and also the workflows that were used to generate those experiments. And what we mean by administrative metadata was all the aspects related to data governance, policies, what kind of, how to define a certain data set, and who are the communities responsible for this, owners basically, who have access to this data. And the last part is the submission spreadsheet, which is probably the most work that we did together in this project, was to come up with a structured form of capturing metadata, ensuring that it fulfills the consistency and completeness of all the information we need as part of research as well as administrative metadata. And also we thought a little bit about how can we ensure that we get submissions which are not, which are complete and which are factually correct. So for this, we ended up designing the model with using ontologies. So for each category that we were capturing as information, we were looking at specific ontologies which can enrich the fields that we are capturing. For example, if you're capturing information about sample and tissue, then all of those information will be annotated with ontology specific to those samples, like Uberon and Brenda for sample. For disease phenotypes, we started using human phenotype, Mondo, and Odo. And for data uses and data access, we started looking at codes to provide permissions for how the data should be used. So these kind of ontologies were used to enrich the model. And for those fields for which we could not enrich, for which there are no ontologies available, Then we started coming up with a list of vocabulary terms like, for example, there is no ontology which describes the different library types in a certain genomic experiment. Then we came up with a checklist of the different terms that can be filled. And this will also be provided to the data submitter, so they don't have to write anything on their own. Everything is in a certain way automated. They can just fill in values according to the information we provide them with. So it also enables them to submit the data much more easily rather than them having to type something on their own. So now I will quickly walk you through the submission process involved in GHG A. So it's a fairly simple process. It has mainly 3 steps. One is when you want to submit data to us, we first ask you to fill in a pre-submission inquiry form with very few questions on what you want to submit with us to see if you're on the right platform to submit the right type of data. And once the submission inquiry is, you've passed that, then you will have to go to the data processing contract phase, where you will be agreeing to the terms and conditions in which you want to submit the data, and what kind of data should be made available, what should not be publicly available. All of that will be decided. And then you prepare for data submission. So when you initiate a certain submission, there will be data stewards who will be guiding you through this entire process. So once you initiate a submission, you first prepare the data steward along with you will help you in preparing the research metadata. And then he will process the administrative metadata. Then once both of them are filled, then you will validate the whole metadata that you have put together, and then it will be submitted to the data portal. So metadata preparation is something that we wanted to ensure that we give, as I told you in the beginning, we wanted to guide the submitters to submitting the process in a more intuitive manner. So what we did was we gave them a structure in which it will look like we call it as the bench to repository approach, where what would a person do when he does an experiment in a lab? So you have an individual from which a sample was extracted, and then it was put through a certain experiment. And then after that, that was subjected to some kind of a secondary analysis. And then you have processed data. We try to give a similar structure even to the research metadata preparation. And therefore, it makes it easier for the users to provide the necessary information. And then with respect to the administrative metadata, we work with the data stewards and we ask them to come up with these contracts and identify the ownership information, how much of data can be provided and not provided, and also prepare contracts with the data submitters. The third part is validation. In a way, it can also be called as curation. What we do is first we have the metadata that we get in the form of an XLS spreadsheet. That will be passed through a transpiler. A transpiler basically converts the existing XLS, whatever comes from the spreadsheet, into a JSON format. And after that, this will be validated against our model, our empty spreadsheet, to see if all the modalities are matching. And if everything matches, there are no syntactic or semantic mishaps, then the metadata report will be generated. And also the metadata JSON file will further be uploaded by the data steward to the data archive through the data stewardship kit that has been put together. So the process is pretty much straightforward, but at the same time, it involves a lot of human intervention as well, sometimes to check for the consistencies or inconsistencies when data is being submitted. So it's a semi-automatic approach, but in the future, we want to make it fully automated. Yeah, so as I said, once you finish this whole, then you process, you basically upload the file to the data portal with the help of a data steward. And then once you publish this, it will be available in the data portal. Of course, you can also have reservations on what kind of data can be publicly available and what should not be publicly available. And all of this will be agreed in the contracts put together. And we also came up with some strong guidelines on, even in our knowledge base, we have put together a strict set of guidelines for us to follow and also for submitters to follow, is to ensure that the data that is submitted is complete, the provenance information is captured, accuracy of information provided, Also to check with consistency and coherence, whether all the values and properties match. The format accessibility and also openness, basically, on how to access the data. So the spreadsheet that I mentioned to you looks like this. It has the field names. It has clear description of what the field entails, what type of value is going to be captured in that field, whether it's multi-values or single value, and also if there are any restrictions, and also whether that particular field information that we are capturing is required, mandatory, or mandatory, conditionally mandatory, or optional. So all of that information is also provided to enable the users to fill in this submission much more easily. As I said about the legal contract, how it works between the controller and the requester. So a controller is the person who is generating the data sets and submitting the data set to the data portal. And the data requester is somebody who wants to make use of this data. So as I told you in the beginning, we have a centralized nub hub, which takes care of all the contracts between the controller and the requester. The first step of this process is to enable a contract between the data controller and the central, the processor of the data. That will be the first step. Then when a data requester comes in with a certain request, then he basically will be contacting the GHG A central. And once he is contacting him, then we will put the controller and the requester together into a conversation where they will come up with agreeing on the terms and conditions on how the data should be used, which is the data transfer agreement. And once data transfer agreement is agreed, then we will go into the approval process of processing that request. And once that request is processed and everybody is happy, then the requester gets access to the data. So this is a structure that we have put together to enable a strong ethico-legal framework for a data provider to provide the data with confidence, and also for the data requester to handle the data with all the conditions in mind, and therefore there will be no misuse of data or misused opportunity from the data that is being provided. I was also asked to touch a little bit about handling sensitive metadata, because we're dealing with human data, so obviously a lot of sensitivity is involved. So the biggest dilemma that we often faced when capturing metadata for this particular project was the clause from GDPR, because there any kind of metadata can be qualified as personal data. So according to this particular law, anything that we are capturing, whatever we call as data about data, is considered personal metadata. So we cannot really capture anything at all. But it also states that personal data related to identifiable living individuals, even if they are only, if the identification is possible through a combination of other factors, that is also considered as personal metadata. So what they mean here is, if say there's a person, you have associated features such as sex, age, ancestry, diagnosis, all of these information, in a combination of this can also help in identifying a patient. So that is seen as a breach. So obviously, we as a platform will never qualify for this. So we came up with various combinations. We came up with various regulations to see how this clause can be, surpassed. And this also put us in a big question on what we are trying to do, because on one hand, we want to capture as much data as possible to ensure that we provide good quality metadata. On the other hand, we are dealing with the problem of privacy and security, so we cannot really capture everything. So we really had to find a balance between the two. Especially in conditions like rare diseases and living in a country like Germany, where the population is not so much, the risk of re-identification is also very, very high. So we had to be extra careful on how we come up with, how to kind of balance both what we want versus what we should be doing. So what we came up with in terms of features, I'll just give you a couple of examples. We did a lot of modifications, but I'll try to just stick to a very high level info. So we started capturing geographical information at a very high level. So whatever information we received from the data provider, we tried to keep it at a very high level in such a way that it does not lead to any kind of re-identification. Say, for example, data access committee was provided with information on where the lab in which this data was sequenced, who was responsible, which university. All of that information will be provided. But when this data is put in a data portal, we will only provide the information that this data is obtained or it's produced in Germany. That's it. We will not add information pertaining to the postcode or which lab it was sequenced. All of that will be completely off the record. So it will be archived, but it will not be visibly available for people to search. The second thing that we try to do is when we are trying to capture age-based information, we came up with bands, like coming up with ranges. So it also prevents the risk of re-identification. Instead of if somebody comes and says, look, yeah, I'm a 38-year-old sample, then we will give a range between 30 and 40. So it also prevents any risks of re-identification. The third one is in terms of capturing diagnostic information. So we have the ICD code dictionary, which gives you the list of different diseases. And each of those codes can have very specific numbers. Say, for example, if you're talking about rare disease, a particular strain of rare disease will have an ID U31.0.1. So what we will do is we will only capture the first 2 digits of the particular disease hierarchy. So it doesn't lead us to saying this is the particular strain because of which this genetic disorder happened. All of that information will not be provided. We will provide a very high level structure and say ICD code 10 is the disease that this particular sample is pertaining to. So we came up with, with clear descriptions. So even when a data submitter comes to us and he submits the spreadsheet or the data on his own, he has to go through these descriptions that will be given for each field. So we came up with these clear descriptions so they don't provide any information that they should not be providing. At the same time, even if they provide, we will ensure that we don't publicly make it visible. So those kind of regulatory checks are being done. In general, what is being captured in this model is basically detailed experimental metadata and aggregated information at an individual level, which prevents us from the risk of re-identification, at the same time capturing data, which makes the data also reproducible for other users when they download this data set. What is not captured is detailed information about a particular data subject. Say, for example, we as a consortium cannot take any liability for information that is being provided, but at the same time, we will ensure that whatever information we get will not be subjected to re-identification. So that's one check we take care of. And second thing is also every possible detail about experiments and analysis performed will be provided by the user, but we will make use of existing checklists such as mixed standards to ensure that the reproducibility is still possible without frustrating the users of GHG A during the submission process. So we also have our own checks to ensure that the data that is coming in will be properly managed. In case of handling clinical data, I think this was something Dicky sir wanted me to talk about, which is something we are still exploring. We have not yet, we don't have a clear answer to this, but we are thinking of integrating a concept called PhenoPacket, which is nothing but a schema designed by a professor in Berlin, which helps in researchers and clinicians to exchange phenotypic clinical-based data in a standardized manner to accelerate research and improve patient care. So this schema, what it does is it captures every diagnostic information possible about a certain patient or a sample. And this information can be further linked to an individual pertaining with all these features. And this schema is also interoperable. It is designed in a similar schema like LinkML that we have. And it captures also the time-based information related to the disease, like age, onset of the disease, progress 6 months, 12 months, all of those information. Longitudinal data is also captured in this model. The schema basically looks like this. It is very, very detailed, and it captures almost everything that you need to know about the clinical interpretations of a particular disease or a modality. How we plan to incorporate a schema like this into our model is basically identify certain modules, which is very, very important for us. Say, for example, information related to, say, measurement or phenotypic feature or interpretation of the disease is very important for us. Then what we will do is we will just identify those concepts, integrate it into a sample file, a supporting file pertaining to a certain sample, and we will encrypt this. And in the data portal, we'll make it visible that we are capturing clinical information, but we will not tell what clinical information are being captured. So we want the data submitters to know that this particular data set contains clinical information, but we will not tell what information it contains, just to prevent the risk of re-identification, or basically not ensuring what details we have in terms of that particular sample. So this is one concept we are currently exploring on integrating. We have not yet implemented, but we are just studying the structure at this point of time. The second aspect that we are also exploring is provenance metadata. So whatever I spoke until now was pertaining to experiments and samples, but we have not really explored a lot about the analysis, like the secondary analysis performed on that particular data set, and how we want to capture, what kind of information we want to capture in terms of analysis. So we are looking at various schemas that already have are designed for capturing analysis metadata, which is like RO-Crate, NF-Prof, are two strong schematas that exist to capture analytical information. So we are currently seeing how to incorporate that into the model that in GHG A. So making it more standardized, and nothing is, basically nothing is, nothing is designed by us, but something that already exists. We just want to place it into our model. As much as all the good things that are happening, there are also a lot of limitations that we are dealing with. I think I've repeatedly mentioned about this, but it's very, very hard to balance the, to create the balance between access and privacy. Mainly because the more and more data types, new data types are coming in. There are also newer risks associated with it. Especially we are still struggling how to capture information related to demographics, social and behavioral determinants of health. If we want to capture that, we still don't know how. Again, phenotypic information at the molecular as well as the clinical level. How do we incorporate data from electronic health records? All of this is still a big, subject of discussion. We are very much aware of all the re-identification risks, but we still have to see how to incorporate all of this into the model. The second one is we wanted to make use of a service provider called Bacon, which is helping in providing a searchability of genomic variant and associated information without compromising on the privacy of a certain data set. This was developed by the GA4GH consortium in the US, but recently it was also subjected to membership inference attack, which basically means that when such a query is integrated into a certain data portal, then any model that is being trained to identify the features behind this search query has the risk of being identified. It's actually possible that a couple of people have tried it and hacked the Beacon API. Therefore, it limited us from incorporating the search feature into the model because of obvious risks. So we are also exploring what other federated search queries we could incorporate to enable search features for users. So that's a current limitation we have to handle. But I don't want to call this whole presentation as a conclusion, but I would see this as an opportunity or a future outlook on how data portals should be designed. One is data portals like GHG A offers, enhanced data quality and integrity platform for doing reliable and reproducible research mainly. And second is such initiatives accelerate research and collaboration, especially making it easier for data sharing and collaboration amongst institutions. Third is it provides the opportunity for innovation in terms of data management tools, because now we are dealing with lots of contracts and data governance related issues. Then coming up with some annotated tools will help in speeding up the process. And of course, this helps us to give the opportunity to reach out to wider communities outside of research. So we are also doing a lot of workshops, webinars, trainings, not just within the research community, but also to the wider community, bringing awareness to the clinicians, to the lawyers, to the patients, which I have not mentioned here, but we are also doing something called a patient initiative, where we are taking patients as stakeholders into this project and taking their views on how to handle data. So those kind of workshops are being done. Of course, it's an opportunity, and hoping that more such projects come in to ensure that we foster research. I also wanted to provide some resources that you could make use of to read more about what we are doing at GHG A. We also have a GitHub page where we are documenting all the model, versioning it, and also the data dictionary I mentioned about. Feel free to take a look at it, and also the submission spreadsheet in case you want to design something like this for your own institution. As I said, we are also doing educational videos in terms of, or educational webinars in terms of research data management. So happy to do courses also for Ramachandra if needed. But we are doing this across universities and institutes here. Basically training wet lab scientists, even bioinformaticians on how to come up with good data management systems within their own labs. Also, I just wanted to say that as an initiative from EMBL, I'm co-hosting a podcast series called Knowledge Catalyst to promote open science in general, on how people from different fields are within EMBL are, what kind of problems they're facing, how they're addressing a certain challenge. All of that is being explored in this podcast. So if you are interested, please do take a look at it. And of course, we also would like to hear some feedback from you. With this, I would really like to say thank you to everyone for joining this this webinar, and I would like to especially thank Dicky sir for having me here, and also to Harsiddhi for for all the back and forth that she had to do with before this webinar. So thank you so much. There were some questions that I have received, which I would like to also answer, but I would like to also see if others have any questions that I could answer now.
Thank you so much, Dr. Anandhi. Now, we would like to open the floor to the participants. If they have any questions, you could just drop them into the chat box to be addressed. I believe, Dr. Anandhi, we have a couple of questions in the chat box right now.
I can take a look at it. Okay. Okay, my question is that using the omics data, what types of omic data are said to be used? As you stated, the components to be used by means of FAIR. If I get your question right, are you asking the different types of omics data that we are currently handling? So far we have single cell genomics data sets, some spatial transcriptomics data set, and just generally NGS sequencing data sets that we have so far captured. Does this answer your question, or? I think it was from Abdul Munafi, so. Okay, I'll wait for him to answer. The next question is, What are the steps to be used or involved of data generation to grant data access in GHGA? So I think I explained the process. It's fairly a simple process. So a data generator, for example, if you're producing the data, you just have to come to us and you have to basically sign a contract with us and say that, yeah, on what conditions you want to you want us to store your data. And in terms of a requester, he also has to come to us and first of all tell us what he wants, what kind of data he's requesting for. And he has to be in an agreement with the data controller, the data provider. They both have to also sign contract with each other to agree. And once the agreement or the exchange has happened successfully, then the data can be requested from our data portal. Then I think another question from Priyadarshini, how can such a data model be applied to countries like India? Would it be different? Such a wide range of clinical data range available in our country, and it would be very useful if such concepts are implemented with our data. That's a very good question. In fact, I think there was also another question that came as part of the Q&A. Certainly, there are lots of initiatives which are being taken already in India. From what I know, I had it answered. I think somebody, just a second. No. I think India had a couple of initiatives. I cannot. Yeah, the Genome India project is already, preparing, a catalog for Indian population, and there's also a rare disease registry by ICMR, which are doing something similar. But I certainly agree that, and also I think IIT has put together a genome atlas for cancer genomics already. But for sure, such initiatives are needed. And I personally think what would really work much better is the NFDI structure I told you. Like here in Germany, there are different research communities, different researchers working on the same field come together on projects like GHGA. As I told you, like we have 6 or 7 data hubs producing data. So, but they all came together in this project to ensure that they submit data to us, right? So I think such nationalized infrastructure we should have such infrastructures in India, where common use cases are discussed, and working groups should be formed to basically discuss this and maybe make a proposal and get funding for it. Okay, how do you deal with legacy data sets that lack complete or structured metadata? Oh yeah, many older genomics data sets. Very, very good question, Abhinand. I don't have an answer to this, but so currently what we are doing is whatever data sets we already have, we are trying to see if we are able to trace back to the owner or the data provider itself, and to really see if we can, if the data is still, valid, or if the data can be, if it has a different version now, or like if the data is, some data they just take it off the system completely because it is either, it cannot be subjected to reuse or for whatever reasons. So we try to reach out to them if possible. But if we are not able to reach, we just archive them. We don't provide search functionality or usability for that particular data set. But this is a big problem. I don't have an answer to this. I think Aishwarya, you raised your hand.
Yeah, hi. I had like a few questions, so I thought typing them might be like a bit hectic for both me and you. You actually answered a few questions like as the presentation progressed, but I still have a few more. So I'm currently in the UAE, and Abu Dhabi is like the government is working on the Malafi portal, which sounds very similar to this, except that it's basically to exchange health data across hospitals, so that if a patient has to be transferred, then they can access the data easily. And they have some, like basically they automate the data requesting process, where you mentioned that you have a data steward who's working on it. are you also working on any way to automate this process, or do you plan to stick with human intervention?
I personally feel a human intervention is really needed, mainly because, yes, automation helps to a large extent, but there are certain situations where I think human intervention will really help. For example, to capture like we had situations where we really had to intervene, and I don't know about the contracts because I'm not dealing with contracts directly, but I can talk to you from the metadata level. Sometimes the syntax and the semantic automations are not really good enough. Sometimes you need the human to also see if what is being, what is being ingested is reliable or not, and then you train the model again with the adapted structure. I feel it should be a regulated process, in the sense it should be an ongoing process where there is constant feedback between the human as well as the system, the validator or the transpiler.
Yeah, I agree. I think it's important to keep humans, even if we do a bit of automation. And I had one more question. So since you're building a database of so much data from both research and from the clinicians, or like you're working on collecting data from the clinician side, are you also working on something that you could basically use this data or like in specific niches to predict better therapies in order to assist the clinicians to make better recommendations for the patients or something like that? Because this is also something that I've seen a few companies here in the UAE working on.
Yeah, that's, I think, very brave step to do. I think we are still not yet there, but I can give you an example of a use case where we are dealing with the insurance agencies, right? Like they are, so I don't know how, in Germany, how it works is you have sequencers, and once your data is sequenced, and those sequences are also, they go through this licensing, this insurance company pays these research institutions who are generating data. So there's a kind of a contract, right? So clinicians, we were thinking at some point we should come up with these agentic bots kind of platform where only a clinician can make use of the data, like a LLM-based agentic model, to be designed to answer specific questions, or even multiple clinicians can work on a similar project. We have thought about it, but the regulations in Germany are quite very high. So I don't think we will get there. These can be experiments that we can do within the, for fun, but it takes a lot to regularize it or to take it to mainstream. So
Okay, so you don't think this is something that is going to happen anytime soon in Germany?
Unfortunately, that's I don't think it's happening.
Okay, that was a really interesting presentation. Thank you so much for your time.
No, thank you so much for your questions. I think there's another question from Abhinand. How does GHG handle the risk of linkage attacks, where it may be possible to combine auxiliary data sets like genomic data to... So this is the point, right? So clinical data,
This has happened in other places, not like the 23andMe breach happened.
Yes. So the risk is there, Abhinand, for sure. The thing is also with, I told you, right? Like when even capturing clinical data itself is a huge risk. But the problem here is we are also not in a position to, we decided, at least as a consortium, that we will not look into the clinical information that comes in. We are going to encrypt, like whatever data that the provider is giving, right? It's going to be encrypted. And whatever data is going to be downloaded, it will be known only to the person who's downloading it. So we as a data, like a service provider, we really have no control of this, so that puts us also in a risk of, being a platform which gives the possibility of this linkage attack. I don't have an answer to this, Abhinav, but that's something we are also thinking about, and we don't have a clear short solution on how to solve this. That's why the clinical part is still in the development. We are at a lot of risk, and we are being very cautious whether to take it forward or not. So, yeah. So Harsiddhi, should I go? Okay, another question from Aditya. A small question, data being dynamic, how and what procedures are adopted to check the validity of these data and the results that stem from these data done by research associates as well as companies. Data being dynamic, how and what procedures are adopted? Exactly, so data being dynamic in the sense, are you saying there are different types of data and how we cater to, as a data platform, to the different types of data? Is that your question?
Yes.
Okay, so that's the thing, right? So the structure that we put together is that we have a, we cannot, of course, say we have a structure for all kinds of data sets. Maybe what we can deal with is the data sets that are currently available here. I mean, what we know as mainstream data. But the model, again, is designed in such a way that it can capture different new data types, but validity will only be done at a very minimal level, at a very high level, because we don't yet have standards to compare them against with. So we will have to only validate it with the minimum standards that we have in place. Of course, if that becomes a mainstream data set at some point of time, then we will have to come up with various checks and balances on whether the data is valid or not valid, personal, non-personal, all of that can be done. But at this point of time, very minimal checks are in place.
Thank you.
Yeah.
If any more participants have questions, you guys can unmute your mics and ask as well. If not, then Dr. Anandhi, I believe you've got some questions in your PPT as well. You can address those.
Yeah, sure. I'll share my screen again. Yeah. Okay. You can see my screen now, or?
Yes, Dr. Anandhi.
Okay. So I think there were really nice questions that came by. You're able to see my screen, no? Yeah, okay. In full screen mode, because earlier there was a problem. So yeah, okay. So the first question was, what safeguards does GHG suggest to prevent bias in AI research, especially when most data sets are male dominant and female specification conditions like PCOS and endometriosis? Again, I had to think a lot for this question, because it is something that is, it does lead to the risk of re-identification. But as far as GHG as a platform is concerned, one is with strong standards, metadata standards, and with the FAIR principles. We are also supporting the, analysis across cohorts, which we are kind of standardizing data in such a way that it is a foundational, it does provide a foundational step for bias assessment. You're already validating, asking those questions. Is this findable? Is this accessible? Is this reusable? We're asking those questions almost on every step. So in case of specific modalities, specific conditions like male dominant, female dominant, then again, the data, this has to be decided at the data contractual level on how we are reviewing that request that comes in, right? Like whether, what is the accessibility or access privy points for specific conditions like this? What is the consent for this? What are the usages with the information that is being provided? And third aspect is also the DAR, right? The data access request. So what information do we provide to the person who wants to make use of this data by preventing the bias? And I personally feel it's also a very personal discussion between the requester and the provider. But at the same time, as a platform, we will ensure that all that we can do is we can only prevent the risks of re-identification and secure data sharing. But the specific situations have to be weighed according to the user specifications, in my understanding. And of course, we also have to audit the models on a regular basis to just ensure that our model is complying to any kind of biases and any kind of error rates, any kind of errors that are being spotted, and just have to be more vigilant with respect to that. I think the second question was, how do you balance openness for research with strict privacy for patients, especially when data is re-identifiable? I think I mentioned. I think we really need to have strong policy frameworks in terms of contracts, and the model design should also be a bit more flexible. So whenever you do see some kind of a risk, you should be able to either reduce the model size or you should be able to expand the model. And of course, a strong balance between quality and quantity is also very, very important to ensure that, that you make the data reusable with no re-identification. The third question was, with rapid changes in AI and analytics, future uses of genomic data are hard to predict. How do you think about future-proof consent? Is broad consent enough, or do you see a need for more dynamic ongoing consent models? I think it's a very good question. I think in the broader scale, a broad consent is already good enough. And we, at least for GHG A, we do have, we do comply to this broad consent in most cases. But the work stream, as I mentioned to you, the ethical work stream, they have come together with a lot of other consent toolkits. As I shown you in the example on the right-hand side, they came up with how data sharing should be done, how de-identification modules can be adapted. This is not just for GHG, but this is for, in general, data sharing practices and consent option modules. So these modular toolkits can also be explored depending on the use case. And also we have a legacy toolkit that was put together, whether the the GDPR consent is sufficient for processing, or whether other restrictions are also needed. So these toolkits have also been put together. I think just to answer that question precisely, broad consent is good enough, but more modular consents will help us to be more specific to the use case that we are dealing with. Yeah. The next one is, this was a very hard question for me because I was just tearing myself apart. Is it ethical to use human genome data to engineer synthetic life forms or improve model organisms? I'm scared to say this, but yes, it is ethically permissible again, but again, it comes with a lot of reservations again, because the biosafety measures need to be considered, and the work has to be clearly defined as to why it's being needed, and what kind of governance, what kind of risk mitigation analysis needs to be in place to ensure that this is this is okay. And we really have to have very high standards, and we should have good enough reasons to having to do this. But I also know that organizations like World Health Organization, UNESCO are also looking at, I recently read an article related to human gene editing. They are already posing threats and how this can be properly regulated, and all of that. So many consortiums are definitely looking into it with suspicion, I would say, and with a lot of caution. So to answer the question, yes, it is ethical, provided the use case is strong enough as to why it's being, why this is needed at the first place. But if I have to answer as a on a personal basis, I would really stay away from this because human data is quite it has its own risks, and as much as possible not to play with it. The next question is, what is the scope of genetics and data analysis in India? What steps should be taken to create an Indian-based database for rare disorders and other prevalent genetic disorders? I think this is also what I spoke of as a question already. There are some nice initiatives already in place. But personally, what I feel is we really need to come up with more national level working groups that are formed to study and to address these issues, because we as a country, I think every state has its own research. It's kind of decentralized in a big way, so we don't even know what the other is doing. So maybe we should come up with more infrastructures, national level infrastructures, study groups, which will bring these communities together and think of a more centralized solution. But I think a good start is already being made by Genome India, Rare Disease Registry, and Indian Genome Variation Consortium as well. But for sure, we need more of such efforts. The next question is, what is the scope of genetics and data analysis? Oh, is it the same question? Yeah, I think it's the same question. I'm sorry. Is there any restrictions or limitations if we use publicly available data set? I don't think there are any restrictions per se, but a lot of caution is needed because many data sets also come with a lot of legal usage conditions. You should always be careful and look for those codes. Like some data sets will have CC-BI, which basically means you should credit the data or like the data producers. And in some cases, you will see CC0 where no attributes are required. So basically, you're okay to use it without any consent. And another code like CC-BY-NC cannot be used for commercial purposes. So you have to be very careful and to and to be sure that you're clear with the legal conditions pertaining to the data set. Then you have to again check for the ethical consideration a bit. Again, for reasons I've spoken enough about. Citations, again, you have to be clear about giving the right citation. And of course, if you're using data sets without citing them, some databases do have high expectations. If you don't, and then that leads to kind of a research misconduct, you have to be careful. And of course, if there are any other legal compliance in place, you should be careful about that as well. Is it possible to personalize medicine only with genomics data? I personally don't think so, because I think genomics alone is not personalized medicine. There are various layers to it, right? Like, there are clinical, phenotype, environmental, lifestyle, so many added attributes to it. So I think, the answer is no. The next question is, what skills should life science students develop to work in data governance and genomic research? I somehow thought of this question in like two ways. I didn't know whether the question was data governance in genomic research or data governance and genomic research, as in they are two different concepts in a way. So of course, I addressed it as, of course, when you want to do genomic studies, you obviously have to, be familiar with all the biological and genomics knowledge, NGS technologies, workflows building, algorithms, building algorithms, pipelines. And as far as data governance is concerned, I think it's very, very subjective too, because I didn't know data governance existed until I got into this project. So there are various courses being offered by Elixir, EOSC live training. I'm a member of EOSC, so if any help needed for a course, I can try to speak to them and help you do the course. They're offering courses on data stewardship, FAIR principles, data management, ethics in clinical data sharing. Then there are GDPR and privacy courses being offered by Coursera and other communities. Then you also have Leiden University is doing responsible data science courses, which I highly recommend. I've done it and it's quite good. Ethics and AI and Big Data by Linux Foundation is also a course you could consider doing to learn about data governance. Oh, I'm sorry. I think these were the main questions. And I think apart from this, there were some other questions on which I was not very clear of. So I'll try to answer them. Research opportunities in Germany, EMBL for Indian students, and how to bridge skill gap for industry and academia specifications as a beginner. What is your view on the scope of bioinformatics and future relevance of genomics? So EMBL offers an international PhD program. We do have a lot of Indian students coming in through that program. So definitely you can look for opportunities. They publish almost every year. Once you fill in the application, you will be invited here to Germany to meet with the professors and to see whether things are you are able to be in a good rapport with him or her, and then you will be given a PhD position. So the steps are pretty straightforward. You can be in touch with me if I come across any opportunity. I can certainly pass on to you. How to bridge the skill gap for industry and academia? Again, I think it depends. You can start doing a lot of internships in various companies. You can also participate in lots of webinars. It gives you the opportunity, like many companies also offer, webinars. You can participate in them, connect with them. Networking helps a lot in this case. So maybe you should consider doing that. my view on the scope of bioinformatics and future relevance of genomics, I think we are in a golden era, I believe. Research is progressing great, and bioinformatics is adding invaluable discoveries to the field. I think we are going good. Another question was with respect to how to connect metadata with different fields of biology. Metadata is not it is not isolated, right? Any data that you produce will have metadata. So if you're taking a picture of somebody, then every picture will have information about that picture. Who was in the picture? What is the picture about? Where was this picture captured? All of this is metadata. So how to connect metadata with different fields of biology? I think the answer is they are basically complementary to each other. So every data will have a metadata. So I hope these were the main questions that were asked. If there are still any other questions, I'm happy to also take a look at them. Yeah, Garima.
Good evening, ma'am.
Good evening.
I am actually currently researching in the field of microbiology and interested in pursuing bioinformatics in future. So I was I wanted to ask you as a beginner, what should I build for my projects, like to build my portfolio in bioinformatics, like to pursue future higher studies or something? What are the best options?
Okay, Garima, I didn't hear you completely. Are you, what is your current background, and you want to work in bioinformatics, right?
Yeah, yes, ma'am.
So what is your current background? What are you doing?
Ma'am, currently I am researching in microbiology and molecular biology.
Okay. So I'm also a microbiologist. So I did a master's at Ramachandra. I think it opened up bioinformatics for me in a big way. So if you really want to study masters, I really would recommend studying here. And then of course you can always do a lot of courses. These days you have so many courses available, right? To just basics in programming, improving your coding skills, introduction to Python, introduction to R. Many institutions are also organizing workshops like that. So please participate in that and improve your coding skills. Are you thinking of moving abroad, or do you want to be in India? And depending on that, I can suggest what you could do.
Pursue my master's from abroad only.
Okay. So see, if you're a microbiologist and you want to pursue your master's abroad, then I would suggest that don't come as a microbiologist. All that I'm trying to say is you come with a little bit of upskilling, of learning a little bit of coding, little bit of programming, little bit of what is pipeline, how to develop a workflow, little bit. Maybe you might not be an expert in them, but at least get familiar with all of this, because most courses which are offered abroad for masters especially, they are not, I wouldn't say they're very hands-on. You will have to learn on your own. So if you come with a little bit of a preparation of, what you're getting into, then it would be easier to adapt. University of Bonn offers courses in molecular biology as well. And I did my master's also here, called Life Science Informatics, which is also a course you could consider if you want to come. But for this course also, you should not come naive. You should not come with zero programming or zero knowledge about bioinformatics. So better to do some pre-prep courses, and then you can come here. I think Germany also offers other courses in bioinformatics as well. I don't have it all on my, right in my mind, but I can, just search about it and I'll let you know. If you want, you can contact me on LinkedIn. I can provide you with more information. Other countries, again, I'm sure the opportunities are the same. There are lots of bioinformatics-based courses. You just have to search and apply for the one you are interested in. Does it answer your question, Garima?
Yes.
So Madhurima asks, I'm curious about the human side of all this data sharing. When patients donate their genomic data, do they really understand what they're signing off? And once the data is stored in an archive like GHG A, do patients have the right to change their mind later and ask for their information? It's a fantastic question, Madhurima, because I did not talk about this in my presentation, but I mentioned it briefly. Patient concerns is something we in the project have included in the sense we are doing a project called Pagoda, which is the patient's perspective of genomic studies. What we're doing basically is we are inviting them, patients who are basically getting treated in the hospitals. We are inviting them to participate in our project workshops. They observe, they listen, and then they ask questions to us on everything they need to know about data. And we ensure that we make them as stakeholders in this project, which means they have a say in whether what they would like to do if their data were being used, how should that be put to use? So it's a good question, but I don't think we have a real solution because it's an initiative that very few organizations are beginning to have. But in most cases, whatever data is being, when a sample is being taken and it's being used for research, they're just informed that their data is being used for research. But most of the situations at times, they're not really agreed up, they don't agree on any conditions, specific conditions on what they should be doing with the data or not. And that's why in this project, we came up with this initiative. And I think that should also be the future of other projects, taking patients as stakeholders into the project and having them involved in every aspect of research. With respect to whether they have the right to change their mind or later, so this I can tell you from the data provider's perspective. I can't tell you from the patient's perspective, because if a data provider, in the sense a person who's generated the data, right, taken the sample from the patient and he's generated the data, for whatever reason, he says that, look, I don't want this data to be part of your portal anymore, then we sign another contract and we take the data set out of the out of GHG A, retaining the metadata alone. Whatever data that they provided in terms of raw data or processed data, that will be completely deleted. The basic metadata information alone will be captured, but in the portal, it will also be mentioned that this data set has been truncated. So nobody can have access to that data. But I can't speak for the patient because I don't think we have the patient's role in this, unfortunately. Does this answer your question? I hope so. Okay. Ma'am, how can genomics data governance create sustainable business opportunities for young researchers or startups in India? Actually, that's it will open up. I think it's a really a new field with respect to especially governance is something, especially life science. For life science, it's relatively, I would still think it's new. I personally think we should invest a bit more on these, like coming up with companies which write contracts and they act as a broker between the researcher and the patient, and they generate like what we do in GHGA, we can independently do also as a company. Where you become, but of course you should be very much aware of the law. We should be aware of the science involved, the politics, everything. You need to have a clearer idea. But I think it would be a good business opportunity to definitely explore in India as well. So can use of blockchain bypass the patient consent and just the data alone for all purpose? I don't think I get the question completely, Aditya. Can you please clarify that for me?
I mean, for instead of typing, I'm just spelling it out across. Like yeah. So what I meant by blockchain here, it is like instead of identifying X, Y, Z patient, if we can through the use of blockchain, which doesn't identify any persons individually, So only if the data are identified individually, we need to get consent from those particular persons. So if we don't do that, then we don't need the patient consent, right?
But how will the blockchain be introduced? Are you saying it should be part of the ecosystem, the data provider ecosystem, or are you saying this should be independent of that? How are you?
It's more of independent.
This needs to be thought through. I didn't think of it in this angle. I will have to think through this. I don't think I have an answer for this.
Okay. Thank you.
I'll think through this and I'll get back to you.
Yes, I hope that is it for the questions. Dr. Anandhi, if you would please be open to it, you could share your email ID in the chat perhaps so that more questions can be posted to you personally because we're exceeding the meeting time. Thank you.
Yeah, sure. I'm giving both my, can I put it on this chat?
Yes, ma'am.
Okay, I'm putting both my official as well as my personal email address. Feel free to write to me.
Yes, I hope we can wind up the session for this. Thank you, Dr. Anandhi. It was a great honor to have you here. We are truly grateful for you to take so much time and effort in preparing and presenting your extensive work here in such an understandable manner. Truly inspirational. Thank you very much, ma'am, for your time and patience. And thank you for coordinating with us for this event today.
Thank you so much, Harsiddhi, also for all the organizing. And again, thanks to Dicky sir and all the participants for being so involved and interactive. Very happy to have presented here and look forward to future opportunities where we can speak again. Thank you so much and wish you a nice evening.
Thank you, ma'am. Thank you so much.